Good morning. In this video we are going to explain the structure of DNA using a 3D model made in Minecraft. But to begin with, what is DNA? Deoxyribonucleic acid, also known by the acronym DNA, is a nucleic acid that contains the genetic instructions used in the development and the functioning of all living organisms and some viruses. It's also responsible for hereditary transmission. The primary function of DNA molecule is long-term storage of information to build other components of the cell, such as proteins or RNA molecules. The segments of DNA that carry the genetic information that cause genes, but other DNA sequences have structural props or take part in regulate the use of the genetic information.
DNA is made up of nucleotides, which in turn are made up of a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base. There are 4 nitrogenous bases, which are adenine, cytosine, guanine, and thymine. These nitrogenous bases follow a sequence according to the gene and the particular action it performs. The DNA strand is made up of a sugar phosphate backbone on the outside, forming 2 helical strands, and on the inside, the nitrogenous base pairs are linked together in the form of rungs.
These base pairs are linked by something called a hydrogen bridge, which pairs the complementary bases to the nucleotide sequence of one of the strands. This is thanks to Jargab's rules that dictate that for every A base there is a T base and vice versa, and for every G base there is a C base and vice versa, which is what forms the complementary bases and is an important part of understanding how the base pairs are joined together to form the double helix.
This structure makes it possible to explain different processes within the cell, such as DNA replication or protein synthesis. DNA replication in eukaryotic cells occurs before the cell division in interphase. For this, the DNA strand is opened and this functions as guide to synthesize 2 complementary strands to each of the strands that were opened, based on the complementary bases that act as guides for the bases to be complemented, so that the DNA is duplicated and ready for the process of cell division.
Another process that is explained with model is protein synthesis. In this process, the DNA gives the orders of which protein should be synthesized according to the gene that is to be expressed. His process has 2 steps: transcription and translation. In the first step, the double-stranded DNA opens and acts when the RNA polymerase selects the gene to be copied into RNA. After selecting the sequence of bases, it copies the DNA information into RNA following the principle of complementary bases, this forming an RNA strands of the specific gene to be transcribed. In the second process, the RNA strand enters the ribosome, and by means of the nucleotide sequences, the ribosome acts to generate the indicated protein according to the gene to be expressed.